My name is Troy Broome, and I am a sophomore pursuing a Bachelor's of Science in Psychology, and I am in Dr. Peng's research lab. Today I will be sharing my experimental design and expected results regarding the anti-nociceptive effects of binaural beats on formalin-induced inflammatory pain. Binaural beats is a form of neural entrainment where two separate frequencies are presented to each ear, producing neural oscillations that match the difference in frequencies. The superior medial olive is a structure in the brainstem that is responsible for this phenomenon. Its primary function is to compare the frequencies between both ears in order to localize sound. For example, if I have 400 Hz presented to my left ear and 410 Hz presented to my right ear, then the superior medial olive will compare the frequencies between the two ears and produce 10 Hz frequency bands and project it throughout the brain. Recent studies indicate that daily exposure to alpha binaural beats reduces chronic and acute pain perception. However, this is based on subjective self-report data. This study takes a more objective approach by recording local field potentials from brain regions involved in both auditory and pain processing. By doing so, we can trace the neural response of binaural beats along the auditory pathway and examine how they influence formalin-induced pain. I will implant a total of 4 electrodes into the rat brain, recording local field potential from 4 separate regions. 2 regions are associated with pain processing. These include the ACC and amygdala, and the other 2 are important for auditory processing. These include the primary auditory cortex and the inferior colliculus. There will be 2 separate groups in the study. The first group will not receive any auditory stimulation. They will only receive a formalin injection in order to establish the nociceptive response in the targeted brain region. The second group will receive the binaural beat stimulation and the formalin injection. They will be exposed to 2 alpha binaural beat conditions, 800 Hz and 900 Hz, both before and after the formalin injection. It is important to note that we only have binaural beats data on humans, and rats hear at a higher frequency range. Therefore, we aren't sure which binaural beat frequency they will respond to the most, which is why we decided to experiment with both 800 Hz in the light green and 900 Hz in the dark green. To ensure that both levels of binaural beats don't influence each other, we put a 10-minute phase with no stimulation between the 2 levels of auditory stimulation. To establish the binaural beat response in rats, we will look at the difference in power of frequency bands in the pre-formalin phase across the 2 groups. Then, to establish how binaural beats influences formalin-induced pain, we will compare the power of the frequency bands across the 2 groups during the second phase of formalin. This is where the formalin response is the highest. There will be a total of 12 Sprague-Dawley rats, 6 in each group. The raw LFP data will be processed by power spectrum analysis through MATLAB. This will break up the raw data into 5 frequency bands: delta, theta, alpha, beta, and gamma. A data point will be collected every 10 seconds and averaged over 5-minute intervals. Each square you see in the experimental design is a total of 2 data points. To analyze the data between the 2 groups, we will utilize multiple ANOVAs in SPSS. Now let's move to our expected results. During the pre-formalin phase, we expect that alpha binaural beats will increase the power of alpha while keeping the power of the other 4 frequency bands the same. This data will confirm the neural entrainment theory of binaural beats and establish its response across the rat brain. When looking at the no auditory group in the second phase of formalin, the power of all of the frequency bands will be high. This indicates the processing of nociceptive signals. When we compare that to the binaural beats group, the power of all of the frequency bands will be low, with the exception of alpha. Because we are producing alpha frequency bands, the power of alpha will increase, followed by a decrease in every other frequency band, possibly indicating a reduction in pain perception. If this hypothesis holds true, it could provide objective neural evidence supporting the anti-nociceptive effects of alpha binaural beat frequencies. Future studies should include a control with 1 pure frequency presented to both ears to isolate the specific effects of binaural beats from general auditory stimulation. In summary, this study uses LFP recordings to explore how binaural beats might modulate pain perception at the neural level. If successful, this research could support non-invasive, low-cost strategies for pain management.